Zhengsen Yu is a PhD student from the Fisheries College, Ocean University of China. And unfortunately, I don't have a short introduction about him. Let's allow himself to introduce himself in the talk. But he was going to present a study on the taxonomy and reproductive biology of the Pacific seaweed pipefish from China, which is a species that have been also studied by another speaker today. So let's give the stage to Zhengsen Yu. Okay, you can go ahead. Please introduce yourself first, because I don't have your introduction, actually. Yeah.
Good afternoon, everyone. I'm Zhengsen Yu from Ocean University of China.
Sorry, guys, we have some problem with the computer. Just a second. Okay, go ahead.
So honored to have an opportunity to have a speech at this meeting. Today, I will introduce a study on a species, Syngnathus schlegeli, taxonomic identification and reproductive biology of this species. I will introduce our work in the following order: introduction to our objects, material and methods, results and discussion, and conclusions. First is a brief introduction of our study object. The species Syngnathus schlegeli. This species belongs to Syngnathus and family Syngnathus and genus Syngnathus. Most of the species of genus Syngnathus are found from the Atlantic Ocean. In China, only 2 species have been recorded. They are Syngnathus arcus and Syngnathus schlegeli. Syngnathus schlegeli was only reported once, while Syngnathus arcus was a very common species in China. However, we found that the Syngnathus arcus in China may be a misidentification. In this study, we first identified the Syngnathus species in China and then studied the reproductive biology of this species. In general, our taxonomic identification was based on morphological and genetic comparisons, and the reproductive biology was studied through biological examination and microsatellite analysis. We identified the Syngnathus species in China. We collected a number of specimens from the coastal water of China. The locations include Dandong, Yantai, Qingdao, and Zhoushan. To identify our specimens, I first examined and counted, measured our specimens to get the morphological date and compared our morphological dates with the previous records. And in genetic analysis, we sequenced the core1 fragment and compared with the data in the internet. And also, we implicated the phylogenetic analysis to help our species identification. The reproductive biology study include fecundity and mating pattern. To study the fecundity, we collected the specimens weekly during April to June in 2013. In total, we collected 1,200 male specimens with eggs, and then these specimens were measured biologically. And next, we counted the number of eggs based on weight ratio method. To study the mating pattern of this species, and we implicated microsatellite analysis. According to our results of morphological comparison, the species in China should be Syngnathus silasigo, but not Syngnathus arcus. The common reports of Syngnathus arcus should be a misidentification. Although the two species are very similar in most morphological characters, we found that the two species can be distinguished from each other by two characters. The first character is the color pattern. Syngnathus schlegeli has a brownish body, and the body usually motivated with pale. But Syngnathus arcus with distinctive body rings, colored sandy brown with darker bars all along its body. The second character is the body length. According to our results of morphological comparison, the slender length of Syngnathus arcus are longer than Syngnathus schlegeli. And furthermore, our genetic analysis was consistent with our morphological results. In the phylogenetic tree, we reconstructed based on core1 sequence. We found that our specimens were clustered with a sequence of Syngnathus schlegeli from Japan, and were separated from Syngnathus arcus. And the genetic distance between the two species is 13.2%. Next is the result and discussion of our reproductive biology. To study the reproductive biology of this species, we first recorded the biology data of these specimens. And next, we divided these specimens into groups according to their length and weight, and counted the eggs of these specimens. According to analysis, the species have a highest absolute fecundity, reaching to 495 and lowest 199. According to analysis, we can see that the fecundity had a positive relation with the body length and weight. To study the mating pattern of the species, we selected 10 pregnant male specimens. And first, we also recorded the biological date of these specimens. And selected 6 polymorphic microsatellite markers to genotype the 10 males and their 360 offspring. According to analysis, we can find that multi-motherhood phenomenon was detected in all the 10 males, and the progeny of each male came from 3 to 7 different females, and the average is 4.3. Among the 10 males, I found that the contribution of female parents of offsprings of the 4 of the males had a significant difference, but the contribution of the female parents of the other 4 female male parents are essentially the same. We suspected that the ovulation of female parents are different at each mating, and the reason may be that the female cannot fully fill the pumps of males, and so they must be supplemented by multiple mating. We suspected that the mating, the ovulation mode can be 3 types. The first type is the females females displace all the mature eggs in the ovary into the male pumps. And second is the female only releases the remaining mature eggs from the last time into the pumps. And there are also may be intersubstance in mating caused by mating competitions and environmental factors. Finally is a brief conclusions of our study. The previous records of Syngnathus arcus from China misidentifications of Syngnathus schlegeli. The absolute number of fertilized eggs in male was 109 to 495, with an average of 263. The absolute fecundity of was directly related to the individual size. The polygynous mating pattern is very common in this species, with an average mating number of 4.3 male per phase. Multiple mating not only improves the fertility of the male parents, but may also enhance the genetic diversity of the offspring. Thank you for listening. That's all.
Thank you for the talk. We can have the question for the talk. Are there any questions in the room? How about the people online? We switch to the online.